each year, a lot of you, about hundreds of you, used to come to CCMB, hear to one of the scientists and discuss with them about their work, and then write a selection test. Based on your performance on that selection test, about 10% of you are selected to come and spend time with us in person for 2 weeks. Ever since COVID started, the first part of the interaction, the inaugural session, basically turned online, and today is our inaugural session for the year 2022. So before we go ahead, I'll introduce you to Director CCMB, Dr. Vinay Nandikuri, and hear about what YIP aims to do. Vinay, over to you.
Yeah. Hello, kids. It's a privilege to have all of you guys come to CCMB and look at what CCMB does. CCMB as an organization, as an institute that works on molecular biology, started way back in 1977. And in 1986, we opened our campus here in Habsiguda. We work on various biological aspects, which cover all kind of basic biology elements. So now biology, if in the last 2 years, I'm sure you guys read in textbooks, and in the last 2 years, you heard a lot about the viral SARS-CoV-2 infections, and then you may have heard about RNA viruses, DNA viruses, and all kinds of things that normally people was not part of the regular vocabulary of people around and across. Even school kids who are, like most of you, have to be away from schools for last 1.5 years because of this pandemic. And our interests are generally, as a scientist, we are part of this particular process of trying to understand how this and many other pathogens and other our own body cells function at the basic level. That's the basic purpose of the kind of research that we do. And so we are interested in understanding things at molecular level. To give you a brief kind of an idea, that basically we all know that DNA is the genetic material, and after DNA you get RNA, and from the RNA we get proteins, correct? So now, how do you visualize this? How do you look at the DNA? You can't see DNA. DNA is very small, but how do you visualize the DNA? How do you actually isolate the DNA from the cells? How do you look at these cells actually, whether they are living or can we actually look at these cells? And how do we investigate these functions of various things that happen inside these cells? That is the purpose of your visit, or your, if any of you who are going to come and work in CCMB, interact with scientists for 2 weeks, are going to understand some of these aspects that I'm talking about at practical level. You can visualize DNA. There are methods for it. You can look at the cells. Of course, there's a microscope is a way for that. And DNA gives RNA. It's also known that RNA gives DNA. So all these exciting facets of science, there are many more things, and there is many more aspects of science that are part of CCMB. And we want students like you to actually be part of this journey to learn, so that you understand and you're enthusiastic, and some of you would become part of this journey in future. So there is a process for selection, which I'm sure you are aware of it. There will be a test and all, and some of you who get selected will spend about 2 weeks with us. During these 2 weeks, you work with our colleagues who are working in various aspects of science, and I really look forward. Basically, I want you guys to take this opportunity to do the best, figure out what are the kind of techniques, what kind of equipments, what kind of things that are used for answering this question. If some of these things interest you, you should pursue science with the purest heart, and maybe, who knows, some of you may end up being future Nobel laureates. All the best. This is a basic message that I want to give you. Look forward to seeing you people at CCMB.
Thank you so much, Vinay. As we move ahead, let me quickly introduce you to our speaker for the public lecture today, and also the topic that he's going to touch upon. Dr. Raghunan Thirumalai is going to be a speaker for the lecture today. Dr. Thirumalai, or Raghu as we call him at CCMB, is of course a scientist, and he studies the bacteria, Mycobacterium tuberculosis, that causes TB. And beyond, hopefully we'll be beyond the pandemic one day, and then we'll again be faced with the truth how tuberculosis is still a huge problem for a country like India, and understanding the biology of this mycobacterium plays a huge role in how we fight about this health crisis. And Dr. Thirumalai is going to touch upon some of those, as well as discuss how drug resistance is a huge problem to healthcare, and particularly in TB, in his talk today. Raghu, over to you.
Hi, good afternoon, everyone. I am really honored and privileged to be part of this program and to be the opening speaker in this really unique program that we've had for several years. And I've been part of it as kind of an instructor in the past, but I thank you to Vinay and Somdata for picking me as a speaker to get things started here. So let me share my full screen. Just tell me if you can see it.
Sure. In the meantime, I should also tell the participants that they can put in their questions in the Q&A box. The chat is disabled, so Q&A box is the only place where your questions can be put.
Can you see my full screen now?
Yes.
Can you?
So I actually see Raghu as the full screen and not the PPT. Why is that, Shrikant?
Shrikant, can you see my PPT?
Okay, I can now. Okay, all good for me.
Yes,
Okay.
I can see you.
Okay, is it moving?
Yeah.
Fantastic. So as Vinay said, today is literally a celebration of science for us. It's wonderful to have all you young science enthusiasts joining us today. And hopefully we'll get to interact physically with some of you when you all come and participate in this program, this 2-week program, hands-on, and work with us in our laboratories. So today we're going to talk about something very, very important. Some of you may be familiar with elements of this, and that is antibiotic resistance. And before I actually go to antibiotic resistance, I'll go through several other basics so that I can bring you up to speed on the subject, and then go to the actual subject itself. So as Vinay said, we are located in Hyderabad, and we are one of the 37 constituent laboratories of CSIR, and we investigate several things, and some of them are listed here. These are our thrust areas. Is my pointer moving?
Yes, Raghu.
Okay. So you can see, like when I said, we investigate different aspects of basic biology. And one of our thrust area is studying microbes and the biology of infection. And that has something to do with today's talk as well. So I am part of the group that studies microbes and biology of infection. And as Somdata said, we look at the pathogenesis of Mycobacterium tuberculosis, the causative agent of TB. But today we're going to talk about not primarily TB, but various other things as well. And before we get started, since we are scientists, budding scientists, science enthusiasts, let me raise a question. What makes a scientist a scientist? Is it just a degree or the term scientist on their door, or is it something else? And if you were all here in person with me, I would show you some photographs and ask you by a show of hands to tell me whether you think any of these people whose photographs I'm going to show you now is a scientist or not. So let's get started. So here's the first photograph. Some of you may recognize him as a great artist and painter, Leonardo da Vinci. And most of us know him as the creator of the Mona Lisa, but he was much, much more than that. So do you think he's a scientist? Okay, think about it. Everyone knows who this is. Probably one of the most recognized photographs of our times. This is obviously Albert Einstein. This photograph was taken on his 72nd birthday at Princeton University. So do you think Einstein is a scientist? I'm guessing many of you will say yes. Here's Madame Curie, one of the only 4 people to have won 2 Nobel Prizes. She won for the discovery of radium and for the discovery of radioactivity. Okay, you think she is a scientist? Here's Mahatma Gandhi looking into a microscope, and what Mahatma Gandhi is looking at is a slide of Mycobacterium leprae, which is a causative organism of leprosy. You think Mahatma Gandhi is a scientist? Here's, I hope most of you recognize this. This is Sir C.V. Raman, who won a Nobel Prize for the Raman effect. Yes, I'm guessing most of you will say Sir C.V. Raman is a scientist. Here's 2 people in a photograph, and this is taken from Google Images. So if you go to Google and type scientist and click on images, this is the first image that will come up. Do you think these 2 people are scientists? Here's a photograph of a movie character called Doc Brown from the Back to the Future series, one of the most brilliant time travel series that has ever been made. Do you think he's a scientist? And here is a cartoon character called Dexter from the series, or the cartoon series Dexter's Laboratory. Do you think he's a scientist? So I think most of you will have differing views on which of these people you think is a scientist. Many of you will say, no, Da Vinci is not, he's an artist. But if you go back and look, Da Vinci is probably one of the greatest scientists ever of all time. He predicted and actually drew diagrams of things like tanks and flying machines and helicopters. Some of you may think Mahatma Gandhi is not a scientist, but I would argue he was one of the greatest social scientists of all times. Okay, so we can argue about who is and who isn't, literally or figuratively, but let's look at some definitions as to who or what qualifies somebody to be called as a scientist. So by definition, this is a definition given by someone called William Weevil in 1833, and he says a scientist is a person engaging in a systematic activity to acquire knowledge that describes and predicts the natural world. This person may be an expert in one or more areas of science. And the third point is an extremely important part of this. Scientists perform research towards a more comprehensive understanding of nature, including physical, mathematical, and social realms. Or you may choose to argue that a scientist may refer to an individual who uses the scientific method. So what is the scientific method? And I summarize here what the scientific method actually comprises. So scientific method is an empirical method of knowledge acquisition, which includes careful observation, and that also the part of this careful observation is rigorous skepticism as to what you see, what you didn't see, what somebody else saw, whether you saw the same thing when you were looking at the same object or the same phenomenon or the same thing. And based on your observation, formulating a hypothesis, and based on the hypothesis, testing this hypothesis using experiment. Based on what data you get out of an experiment, you would see if the deduction from the experiment actually is consistent with the hypothesis you made. And then you would continue to refine the hypothesis based on the experimental findings and then repeat the whole process. So in principle, any one of us who uses this thought process to analyze a particular phenomenon, a particular situation, or a particular problem should qualify as a scientist, including all of you in the audience. So if somebody asks you a question, if you start thinking about this question using the scientific method, you would qualify to be a scientist. You needn't necessarily have a degree or have a position in a research institute to be a scientist. So this is some food for thought for you. If you go back home, you can think more about it and read more about some of the people whose photographs I've shown you in the first slide. Okay. So before I actually go on to the subject of drug resistance, I'm going to introduce to you the concept of pathogenesis, because we're talking about pathogens or agents that cause infection. So what exactly is a pathogen? A pathogen is an infectious agent that causes disease or illness to its host. And the word is described from Greek, and actually means that which produces suffering. And since we're going to talk about microbial pathogenesis today, what is microbial pathogenesis? It is what refers to disease caused by the 4 different classes of microbial pathogens that we know: bacteria, fungi, parasites, and viruses. And please be clear that these are completely different classes of organisms. They all have their own unique identity and their unique features. And the only thing that perhaps overlaps is facets of the way they cause infections in their human or other animal hosts. Okay, so since today's session is going to be about bacteria, let me quickly introduce you to what bacteria are. Many of you know what these are, but this is a quick refresher on what bacteria are. They're unicellular organisms. They are prokaryotes, meaning they don't have a true nucleus, and they also lack membrane-bound organelles. They come in many shapes and sizes, and they have varying metabolic properties. And I'll show you some pictures here. These are different kinds of bacteria that we know about. They could be circular, rod-shaped, curved, and have other shapes. They form different kinds of colonies on solid agar plates, and this is how we study bacteria in the laboratory. We can culture them on different kinds of media, including solid media like agar, or liquid media like different kinds of broths with nutrients in them. And one of the main features that we use to characterize or study or categorize bacteria is on the basis of a stain developed by a person called Christian Graham many decades ago. And Graham devised a staining procedure to identify different classes of bacteria which have different envelopes or different membrane structures. And there are 2 kinds of them. They're called Gram positive and Gram negative. So this is the only introduction I will give to you in terms of the diversity and the different kinds of bacteria. And we'll go on to more specific things as the talk goes along. And things will become slightly more complex as we go along. And don't worry if you don't understand some of the jargon I have listed or some of the points I have listed. We can always discuss this at the end of the talk. If we were doing a live session, you would raise your hand and ask me a question. But for the sake of logistics, we have left all the questions towards the end. Okay. So as in everything, there is a good and the not so good. So bacteria can also be of 2 kinds, good and not so good. Sometimes not so good can be called bad. So what are these good bacteria and what makes them good? We know that in the human body, there are about 30 trillion cells. So 10 to the 12 is a trillion. So there are about 3 into 10 to the 3 cells in our body, and that is an estimate. And amazingly, it is estimated that there are about 3 to 10-fold that number of bacteria in our body. And until a few years ago, we had no clue that this was true. And based on a large number of studies over the past 15 to 20 years, including a technique called metagenomics, which identifies all the different kinds of organisms that live in different parts of your body based on genome sequencing and DNA analysis, it has been estimated that there are more bacteria in our bodies than there are human cells. And I show you here that there are bacteria in different parts of your body. Could be in your nose, in your mouth, in your teeth, in your gut, in your reproductive tract. And I list here, in pink are the number of estimated organisms, and in blue are the estimated number of genes that all these organisms actually encode. And if you look at the number of genes that all these organisms or all these bacteria in our body actually encode, these are 100 times more than the genes encoded by the human genome. So there's a huge genome complexity in the bacteria that actually inhabit our bodies. And we know now, based on these metagenomic analysis, that these are dominated by anaerobic bacteria. But why do we call them good bacteria? We call them good because they assist in digestion, They control our hunger, they control our body weight, they are responsible for several mood-related issues, anxiety, mood, cognition, and pain. They control our immune responses, and one of the most important things they do is they synthesize vitamins. Okay, so where do these bacteria come from? Most of these bacteria are acquired when we are children, soon after we are born. When we exit the reproductive tract of our mothers, we take in the bacteria that are present in the reproductive tract, and over the period of the next few years of our early growth, these are the bacteria that we acquire by various ways. One of those can be, animals or dogs licking us. Many of us have done this before. We sit in grass, or we eat dirt, or we just pick up stuff and put them in our mouths. So this is not actually funny. It may appear funny to you, but this is how we acquire a lot of the bacteria that are present in our bodies. And this is how we actually build our immunities. And there is a concept called the hygiene hypothesis, which some of you can go back and read. And hygiene hypothesis states that the cleaner we are as little infants, the less repertoire of immune responses we are likely to have. So the more dirt we are exposed to, the more we are protected against pathogens. And in the initial phase of the pandemic, this was one of the theories to suggest or to explain why Indians were not getting so sick with COVID-19, but that is now a little bit in question because of the third wave, etc. Second and third wave, impending third wave, whatever that might be. For the second wave, which was pretty devastating, we didn't expect it. We thought that we were protected because we had immunity based on our exposure to microbes. But this is not something we're going to talk about. We're going to talk about pathogenic bacteria. Okay, and what are pathogenic bacteria? These are agents of a number of diseases that you'll all be familiar with that have been responsible over the course of history for millions of deaths. Tuberculosis, cholera, diphtheria, plague, pneumonia, syphilis, gonorrhea, meningitis, anthrax. And I show here pictures of agents that cause all these diseases: MTB, Vibrio, Corynebacterium, Yersinia, there's Treponema, Neisseria, and Bacillus anthracis. So they're all pretty looking pictures, but these are agents that can potentially kill you. Okay. Now, what we must also know is, in the context of our discussion about pathogenic bacteria, what we know is that humans have very powerful immune systems. And some of you may be familiar with it. Our immune system has two components: the innate immune system and the adaptive immune system. And both these components make us immune or protect us from basically anything that can make us sick. So the immune system keeps us well, and the fact that most of us are well or healthy all the time means that the immune system is extremely efficient. But ever so often we get sick. Why is that? And we get sick because of the microbes that I've shown you in these pictures. Plus also you could get sick with viral infections, fungal infections, or parasitic infections. And why do we get sick despite having such a powerful immune system? It is because these microbes or these pathogens have figured out how to evade immune responses and still cause infection, okay, which is why they are extremely dangerous. And whenever the immune system cannot take care of these microbes or these pathogenic bacteria, we have a problem. And what do we do as of today when we have a bacterial infection, or we have any kind of infection? We go to a doctor, and the doctor prescribes a class of medicines called antibiotics. Okay, so this is the first slide on the actual topic that we're going to study or talk about today: antibiotics. Okay, so we're going to talk about antibiotic resistance. So before we talk about antibiotic resistance, we need to know what antibiotics are. So the term antibiotic actually means opposing life, antibiotic. Okay, and it is defined as a type of antimicrobial substance active against bacteria. Okay, please note this: antibiotics are active against only bacteria and not fungi, not viruses, not parasites. Okay. For those agents, you have antivirals, you have antifungals and antiparasitic drugs. But when you say antibiotics, it only refers to an antimicrobial substance active against bacteria. Please remember that, and please clarify the doubts of anyone who may think antibiotics can kill other kinds of microbial pathogens as well. So until the early 20th century, we didn't know what antibiotics were, and such antimicrobial substances were not known. It was only in 1909 when a person called Paul Ehrlich synthesized something called arsphenamine. The trade name for that is salvarsan. And Paul Ehrlich discovered that salvarsan, which is actually a dye, has anti-syphilis activity. So that is the earliest record of any antimicrobial substance. And this happened in 1909. Okay. The next discovery happened in 1928 when Sir Alexander Fleming discovered a substance called penicillin. And there's this big story, very interesting story about how Alexander Fleming discovered penicillin. He left agar plates containing a microbe or a bacterium called Staphylococcus aureus near the window of his laboratory, and the agar plate was left open. And when he came back, some of the bacteria in that plate or in that agar petri dish seemed to have died because there was what we call a zone of clearing. And what he noticed was around that zone of clearing, there appeared to be growing another organism. And he identified that to be a fungus called Penicillium notatum. And he extracted an anti-Staphylococcus aureus compound, and he called it penicillin, and the rest is history. Chronologically, the next so-called antibiotic that was discovered was by a person called Gerhard Domack, a German bacteriologist who synthesized something called sulfonamide. This led to the class of drugs called sulfonamides, and he called it Prontosil. And he famously treated his daughter who had streptococcal infection and miraculously saved her arm from amputation. So some of you who are interested in such historical stories should go back and read about this. Next came Salman Waxman, who in 1944, along with his graduate student, Albert Schatz, isolated a compound which they called streptomycin from a streptomycete called Streptomyces griseus, and they discovered that it had activity against Mycobacterium tuberculosis, the causative organism of TB. And Salman Waxman was the person who coined the term antibiotic. Okay, so this is a bit of history for you. We've come a long way since then. This led to a huge deluge in the discovery of antibiotics, either discovery from natural sources or these were created by synthetic organic chemistry approaches. And today we have a large number of antibiotics which belong to various classes. And I've listed some of the classes of antibiotics here: penicillins, sulfonamides, aminoglycosides, tetracyclines, macrolides, rifamycins, glycopeptides, and quinolones. And I've listed these chronologically so you can see how these different classes of compounds were actually discovered. And this was the 30s and the 40s and the 50s were the golden period of antibiotic discovery. And that golden period actually ended by the late 50s. And since then, the antibiotic discovery or synthesis pipeline has literally dried up. So that is becoming a huge concern. And I will come to the reason why that is a concern. Okay. What you have to see here is that when we discovered these antibiotics, eventually we also discovered how they killed bacteria. And we discovered they hit 5 different aspects of bacterial survival. One was cell wall synthesis, one was metabolism, protein synthesis, RNA synthesis, and DNA synthesis. So largely, most antibiotics hit one or more of these processes, which lead to eventually the death of bacteria. Okay. Now, where we have antibiotics, we have resistance. Okay. So what is actually resistance? Resistance is the phenomenon where bacteria become resistant to the antibiotic. And if they are resistant to more than one of these antibiotics, they are nicknamed superbugs. Now, why is that a problem? That is a problem because the infection they cause are harder to treat than those caused by non-resistant bacteria. And antibiotic resistance leads to higher medical costs, prolonged hospital stays, and increased mortality. So why did antibiotic resistance actually arise? And one of the major causes of this has been the misuse and overuse of antibiotics over the years, as well as poor infection prevention and control. And as early as 1941, Alexander Fleming himself, who actually discovered the first natural antibiotic, said this, and I'm going to read from what he said. He said, "It is not difficult to make microbes resistant to penicillin in the laboratory by exposing them to concentrations not sufficient to kill them. And the same thing has occasionally happened in the body. The time may come when penicillin can be bought by anyone in the shops. Then there is a danger that the ignorant man may easily underdose himself and by exposing his microbes to non-lethal quantities of the drug, make them resistant." So you can see how Alexander Fleming, who discovered penicillin, actually thought that this was going to be a real problem in the future. And this was even before penicillin was released into the market. So you can see how visionary these people were. They not only discovered or made such discoveries, but they also could see into the future. Unfortunately, what Alexander Fleming has come true in a big way and in a way that we are finding very difficult to handle. There are many, many different classes of bacterial pathogens that have become drug resistant and become very difficult to treat, including the ones that cause tuberculosis, salmonella, and various other diseases that I've already listed. And the WHO has specifically listed a class of pathogens called escape pathogens to be the next big problem. And these are already causing trouble. Okay, so what are these escape pathogens? These are the major cause of life-threatening hospital-acquired infections. We also call them nosocomial infections, and they have increased resistance to commonly used antibiotics, and they're resistant to multiple antibiotics. So they have been nicknamed superbugs, and they're all, the 6 of them are listed here, and they're called escape because the word escape is taken from the first letter of each of these bacteria. And in addition to that, Mycobacterium tuberculosis, the causative agent of TB, which my lab and Vinay's lab both work on, has become a huge crisis because you can have multiple drug-resistant strains of TB as well as extensively drug-resistant strains of TB. So basically, we have a problem on our hands. It's not a clinical problem. It is a huge public health problem. So this figure shows you the timeline of resistance appearance after an antibiotic was released into the market. And you can see here on the bottom, I have listed all the different classes of antibiotics in different colors chronologically. And on the top of this bar are various clinical isolates of some of the bacterial pathogens that I have listed. And this shows you that each of these pathogens has clinically become resistant very soon compared to as a function of when the antibiotic was introduced into the market. So you could ask, how does this happen? Is this magic? We've released this antibiotic into the market, people start taking it, and very soon, within a matter of decades, you see clinical isolates of these various bacterial pathogens which have become resistant to each of these drugs that have been introduced into the market to treat or kill them. So how did this happen? How does antibiotic resistance actually arise? Is it magic, or is it something to do with the metabolic process that goes on in bacteria? Before I tell you what that is, I'm going to tell you a story about a very special person. Some of you may know who this person is. Here's a picture of him when he was young. If I just showed you this picture, you may not recognize him, but if I showed you this, most of you will tell me this is Charles Darwin. Okay, so this is a picture of Charles Darwin when he was young. Unfortunately, we only see pictures like this when he was old and gray. But Charles Darwin is a legend in our field, in the field of natural science, ecology, evolution, and I'll tell you what he has to do with antibiotic resistance. So some of you may know that when he was around 22 years old, Darwin went on this voyage on this ship called the HMS Beagle. A side story is Darwin was actually enrolled in medical school, but he didn't want to be a doctor. In his heart, he was a naturalist. He just didn't like being in the medical profession, so he quit medical school. And with the help of one of his uncles, Osiah Wedgwood, he got to go on this voyage of the HMS Beagle, whose mission it was to circumnavigate the world over a period of 5 years, and actually do geographical mapping of the coast of South America, and also sail around the world, coming down to Australia and then go around to the Cape of Good Hope, and then go back to Plymouth where they first started. Now, why am I telling you so much about this voyage, and why is it important to this discussion we are having on antibiotic resistance? So while he was on this voyage, Darwin's primary work on this ship was to stop off at these various points, collect samples of plants, animals, make records of biodiversity, and just make general observations. And since he was a naturalist and a scientist at heart, he didn't just make documentary observations. He also started to ask questions on the observations he had made. And one class of observations that he had made was on these birds that some of you may know as finches. So these are now called Darwin's finches. And Darwin noticed that each of these different types of finches had different kinds of beaks. And eventually he discovered that each beak was specialized to the kind of food that these finches were eating. This is just one of the observations he made out of hundreds of thousands of observations and the samples that he collected. And based on all his studies and observations over these 5 years, Darwin did a lot of thinking, and to cut a long story short, Darwin proposed the theory of natural selection. And until that time, nobody really thought about this subject, as to how evolution was occurring, how do species originate, what species originates from what source, what is the concept of common ancestors. And this is something that Darwin's theory answered in a big way. And in 1859, he published a celebrated book called The Origin of Species. And there he proposed that in a population, variants accumulate for various reasons, primarily because of the mistakes in DNA replication, which leads to variants accumulating in terms of phenotype and genotype. And once variants accumulate based on a selection pressure imposed by the environment, only a small subset of those variants survive. And he called that survival of the fittest. And that was because only the fittest under that selection pressure survived. So what does all of this have to do with antibiotic resistance? This is what it has to do with. So the emergence of resistance has everything to do with this concept of natural selection that Darwin proposed. Okay, and I'll tell you how this works. So here you have, let's say, a pool of bacteria. Bacteria will multiply, and sometimes variants will accumulate when the bacteria multiplies, either because there were mutations during the process of copying of DNA, or the bacteria had acquired additional DNA fragments in terms of plasmids from the environment or from other bacteria in their vicinity, or the bacteria may have acquired DNA from bacteriophages, which are viruses that infect bacteria and integrate parts of their DNA into the bacterial genome. So what you will get is a set of variants. and I have listed, or I've showed you these variants in different colors. Okay, so far so good. Bacteria have undergone one round of replication, and they have accumulated variants based on various possibilities, either because of mutations in their genome or by acquisition of extra chromosomal elements that we call plasmids, or additional DNA from phages or transposon elements, which are jumping DNA mobile elements. So you have these variants. Now, if this pool of variants is grown in the absence of selection, all of them are going to grow equally. In one round of replication, you'll have 8, and in the next round, you will have 16. But if this variant is exposed to a selection pressure, and here the selection pressure is an antibiotic, only the bacterium that is able to survive under this antibiotic pressure will be selected. So there's your selection pressure, and there's your survival of the fittest from this population. And over a period of 3 cycles, this 1 will become 2, 4, and 8. And this pool of bacteria that has come as an outcome of antibiotic selection pressure is now resistant to the bacterium. So this is how the theory of natural selection is applied in this context of emergence of antibiotic resistance, where the fittest from a pool of variant is chosen or survives based on a selection pressure. In the absence of selection, you must realize that there is no difference in their fitness. All these different colors of bacteria have equal survival possibilities. But under the selection pressure of an antibiotic, only the bacterium that is able to survive or able to tolerate or able to avoid being killed is going to be survived. Okay, and how did it survive? Because something happened at this step. The mutation allowed it to have an additional function that allowed it to survive in the presence of this antibiotic, or it acquired an element that encodes antibiotic resistance from another source, or it acquired a sequence of DNA from another source, which allowed it to survive this antibiotic pressure. So this is how natural selection is operating at the level of the bacterial population, leading to the emergence of antibiotic resistance. So over the years, we've learned how to study resistance, how to culture these organisms, how to identify the molecular basis of this resistance. And we know now that bacteria apply different pathways or different mechanisms to become resistant to different classes of antibiotic. And these fall under 2 different categories. One is at the level of antibiotic, and the second at the level of the target. I told you that most of the antibiotics that we use today target 5 different facets of the bacterial metabolic pathways: DNA, RNA, cell wall, metabolic processes. Okay, so either the target is affected or the antibiotic is affected. And this diagram shows you that the antibiotic could be cleaved by an enzyme on the cell wall surface, or it could be inactivated by an enzyme when the antibiotic goes in, or the antibiotic could be pumped out by a class of proteins called efflux pumps, or the membrane or the envelope itself could undergo changes. So you have decreased uptake of this antibiotic, which will obviously make it resistant, or you have alterations to its target, whether it is DNA or RNA or a protein, or if the antibiotic is targeting an enzymatic pathway, the bacteria could develop an alternate enzyme to do the same thing. So what you're seeing here is, because of the enormous capacity of bacteria to develop variants, and by the process of selection of those variants under different selection pressures of different kinds of antibiotics, you get these different kinds of mechanisms of resistance. And you may ask, why is it that bacteria are so adept at this? It's because a bacterium can typically divide every 20 minutes. as compared to human beings, which on an average replicate every 65 to 70 years. Okay. So you can see that from a small pool of bacteria, if a bacterium replicates or divides every 20 minutes, in a matter of a few generations, you can generate a huge pool of bacteria. And you can see, you know, from what I told you in the previous slide, that you have this enormous pool of variants that now undergoes selection under a specific selection pressure, which is the antibiotic that you apply to it. Okay. This is how antibiotics have evolved these various strategies of resistance to these multiple classes of antibiotics that we have. Now, obviously, we have a problem and we can't sit tight. How do we deal with this problem? So how are we going to resist this entire phenomenon of antibiotic resistance? And the way we're going to do it, or the way we have been trying to do it, is first identify the mechanisms of resistance. Second would be to populate the antibiotic pipeline, which means let's make more antibiotics, let's make different classes of antibiotics. Let's isolate or synthesize new molecules, or let's get these active molecules from these large libraries of compounds that we may have synthesized in laboratories or in industry. So just like you have libraries which have books, we have compound libraries which may have been synthesized for various purposes by sophisticated organic chemistry approaches. And finally, can we find new targets or new approaches to treat infections? If we don't do this, we are going to be in trouble. So first we could ask, what is the origin of these resistant elements? Are these modern, or do they predate the use of antibiotics? Did they evolve only after antibiotics were introduced into the human population? And the answer is no. Antibiotic resistance is ancient. And I'll show you a couple of studies which actually showed this. This group of Jerry Wright took these permafrost sediments from Dawson City in Alaska, and they recovered DNA sequences from these permafrost sediments by doing what I told you in the beginning, metagenomic analysis, which means you take the sample that you've isolated, isolate DNA from there, and do DNA analysis and genome sequencing. And what they found was that these approximately 30,000-year-old samples actually had antibiotic resistance related elements. So clearly, antibiotic resistance predates the introduction of antibiotics, which only happened in the early 20th century. A second paper identified a sample taken from a cave called Lechigila Cave. This is in New Mexico in the United States. And this cave has been isolated from the surface for over 4 million years. Okay, the previous sample that I talked about is about 30,000 years old. But here is a sample that was taken from a cave which is over 4 million years old, with no animals or metazoans living in this environment. And from this sample, this group of Jerry Wright again identified a bacterium called Pinibacillus, which had a huge number of antibiotic resistance related elements, which they discovered after their entire sequencing and bioinformatics analysis. And when they cultured this organism, they found that it was resistant to 26 of the 40 antibiotics they had tested. This is incredible because this is a place which has been completely isolated from humans or any metazoans. But here's a bacterium that has all these antibiotic resistance elements, which allow it to be resistant against literally 3/4 of all the antibiotics that were tested. Okay, so this tells you that antibiotic resistance is ancient, and you can ask, where did this come from? The answer is simple. Okay, antibiotics, as the word suggests, are actually molecules that are produced by bacteria to fight with bacteria that are present in their environment. So there is a war going on in the natural environment as well, in water, in air, in soil. So every bacterium there or every organism there is trying to survive. And the way that survival works is you survive if you can either survive something that your neighboring bacteria has produced or avoid killing, or just kill that bacteria using a substance that you produced. So the theory is that all of these elements have evolved in these bacteria because they are involved in the survival of these bacteria in their natural environments, and they are involved in a bacterial war. So all these compounds that we have either synthesized or isolated from natural sources are essentially compounds that bacteria use to fight against one another. So it's not surprising to know that this is an ancient element or ancient phenomenon that we are only just discovering. How do you identify resistance loci? And I'll just quickly tell you about some work that we did in our lab a few years ago. And we were in the lab, we study resistance of mycobacteria to known mycobacterial drugs. Okay? And we were interested in a drug called isoniazid, which is a first-line anti-TB drug. And I told you that multi-drug resistance is a huge problem today. And one of the characteristics of multi-drug resistant MTB is that these are resistant to isoniazid. Okay? So we wanted to try to see what the mechanisms of resistance to isoniazid are. So we carried out a process called transposon mutagenesis. We selected mutants resistant to isoniazid, just like I told you in the slide that showed the process of natural selection. We isolated a bunch of resistant mutants, mapped to see where the transposon was sitting, and we ended up identifying 5 new resistance associated genes. And I show you one result from our experiment. So here is what we call an antibiotic susceptibility test. This is a strip that has an antibiotic in it, and here it is isoniazid. So when you put this strip on a lawn of Mycobacterium smegmatis, you will see a zone of clearing, and this zone of clearing represents the sensitive bacteria, which are sensitive to isoniazid present in this strip. Now the mutants that we isolated are this, and this do not show this zone of clearing, which means that they are resistant. So we are now in the process of trying to figure out how these genes actually contribute to antibiotic resistance. And the take-home message from this is that if we can identify mechanisms of resistance, then we can try to devise methodologies by which we can overcome this resistance. We know that most antibiotics produced by screening soil microorganisms have already been identified. And by the early 60s, these were already overmined. And literally the entire spectrum of Streptomyces species has been mapped or mined to identify antibiotics and produce them both at the lab scale and the industrial scale. And we pretty much run out of sources to isolate new antibiotics from. And synthetic approaches are unable to replace this platform because, as you know, nature is irreplaceable. The kinds of variants that nature can create cannot be created on the lab bench. We know now that almost 99% of all species of bacteria in the external environment cannot be cultured, which means that we cannot grow them in a test tube, which means we cannot study their metabolism or try to identify if they're producing metabolites that have antibacterial activity. Okay? So this group in 2015 figured out how to overcome this problem. So they figured out how to actually culture unculturable bacteria. So they devised a device called an iChip10. It seems that now all cool devices have to have this I in front of them. But this is a diagram of this chip. basically, to cut a long story short, what they're able to do is using this device or this chip, they can culture bacteria that are unculturable. And essentially the way they're doing this is to grow bacteria in their native environment, which is in the soil. So they can stick these chips into the soil and culture unculturable bacteria. And what did they get by doing this? They took extracts from 10,000 isolates, which they grew using this method, and they identified a new bacterium which produced a metabolite called texobactin. This is the structure of it. And texobactin turns out has broad spectrum activity against a huge number of pathogens, including Staph aureus, Pseudomonas, E. coli, and Streptococci. And this turns out to be a cell wall inhibitor. Okay. And what we learn from this is there are many, many more such gold mines out there in the soil, which are an untapped source for antibiotics. So this is just the beginning. And if we come up with novel ways, we'll be able to culture these unculturable bacteria and try to find new antibiotics. We could look in unusual places and not just soil or water. And this group looked in an extremely unusual place, and that was to look at symbionts in the intestine of nematodes that live in mosquitoes. Okay, so something called entomopathogenic nematodes, which are basically microscopic worms that live in the guts of insects. So one of these, Photorhabdus canii, led to the identification of something called darobactin, which turned out to be a novel antibiotic, and this had activity against E. coli and Klebsiella, including drug-resistant isolates. So nobody would have thought of looking in an extremely unusual place, that to a bacterium that lives in a nematode which lives inside an insect. There you go again, another unusual place. Can you use non-biological methods to find novel antibacterials? And here's a method using artificial intelligence. And some of you will know much more about this than I do. And this is an incredibly interesting study done in the Broad Institute, where they used a deep learning approach to finding antibiotics or discovering new antibiotics. And to cut a long story short, they used a pipeline where they would take a set of molecules, run a training set, actually teach the artificial intelligence system to identify compounds that may have antibacterial activity. And that led to the identification of a novel compound called halicin, which had broad spectrum antibiotic resistance, antibiotic activity. Okay. What I have shown you here is unusual approaches to finding antibiotics. Okay. What I've also shown you here is you can work on solving biological problems even though you may not be an actual biologist. Okay. So all these examples that I have cited is work done not only from biologists, but they involve engineers. So an engineer actually created the iChip. They involve chemists who actually synthesize molecules. They involve people who have computational experience, because a huge amount of computational biology is required for this. So suffice it to say, you don't have to be a biologist to solve problems in biology. All you need to do is to be able to want to solve a problem, and gone are the days when you had to be a biologist to actually study biology. Those walls are broken, and now you could be a physicist or a chemist or a computer scientist or an engineer. All of you with such skill sets can solve biological problems. So that's a huge development in the field. The walls are literally down. So biology is for everyone, and I strongly urge you all to try and look at biological problems using the aspect that you might be interested in. I will show you two final slides and then conclude. So a new approach of treating infections is from something we called host-directed therapy. And this is an unusual approach because you are not actually targeting the bacterium to treat this infection, but you're actually targeting the host. And why is that important, or why is that useful? It's because using this host-directed therapy, you can actually cut out the whole aspect of antibiotic resistance, because the bacteria is not involved in this. It's the host that is involved in this. And I'll give you one example of work done from the lab of Vinay, who spoke to you at the beginning of the session. And Vinay's lab works on tuberculosis, and they study various aspects of the immunopathology and the physiology of tuberculosis. And they did this really nice work, which was published last year, where they identified a possible host target as a therapeutic target against tuberculosis. So they discovered that the infection of MTB upregulates a protein called SIR2. Don't worry about what this does, but this is a class of proteins called epigenetic modulators. And what MTB does on infection is it upregulates this protein, which benefits its growth. So Vinay's lab used an inhibitor to try and treat this infection and modeled this in a mouse. So they infected mice with MTB, treated those mice with an inhibitor of SIR2, and they discovered less bacteria, they discovered less disease pathology, and they discovered increased MTB-specific protective immune responses. And they also discovered that this compound enhanced the efficacy of isoniazid, which I already told you about, which is an anti-TB drug. So here you have a target which is not a bacterial target. This is a host target. So what you're doing is essentially buttressing the host response to allow it to clear a bacterial infection. So here's a potential target for host-directed anti-TB therapy. And this is a brilliant proof of concept to show you that host-directed therapy could be a reality tomorrow, not very far in the future. And finally, there's this whole concept of phage therapy. Some of you may be familiar with it. And phage therapy involves trying to treat infections with these entities called bacteriophages. And I told you a few slides ago that bacteriophages are viruses that infect bacteria. They can infect bacteria. They could either be what we call lysogenic, or they could be lytic. So lysogenic phages will stay quiet in the bacterium. They'll do nothing to it. They will stay integrated in the bacterium. Lytic phages actually lyse bacteria and kill them. Okay. So this has been actually tried very recently for a patient with an infection with a mycobacterium species called Mycobacterium abscessus. This is a 15-year-old patient with a condition called cystic fibrosis, and she had a disseminated Mycobacterium abscessus infection, which had failed all kinds of antibiotic therapy. And this group screened a collection of greater than 10,000 mycobacteriophages and identified 3 phages that had specific activity against this specific strain of Mycobacterium abscessus. And the patient was treated with a 3-phage cocktail following a lung transplantation, which was necessary because she had very severe cystic fibrosis. And after this treatment, they found that there was significant improvement in her condition, including healing of the wound, skin lesions, improvement of lung and liver function, and weight increase. And this is the first therapeutic use of a phage for a human mycobacterial infection. So there you are. You have these unusual approaches that could potentially become therapies of tomorrow. Today, they are still in either the laboratory or preclinical stages of infection, but these are real approaches that could help us fight this entire scourge of antibiotic resistance. Okay, so what can we as individuals do to prevent or control antimicrobial resistance, specifically antibacterial resistance? And I've taken this from a WHO advisory. Only use antibiotics when prescribed by a certified health professional. If you get sick, just don't go to the pharmacy and ask for an antibiotic. One is that is not the correct thing to do. Second, the pharmacist is not even supposed to give you this unless you have a prescription. And you will see that antibiotics all have a red stripe on them. It means you cannot get them or should not get them without a prescription. Never demand an antibiotic if your doctor or health worker says you don't need them. Never share or use leftover antibiotics just because you think it might be useful to you. Most importantly, practice good personal hygiene, both at the personal level as well as when you're preparing food. Choose foods that have been produced without the use of antibiotics, which are used extensively where animal husbandry is concerned, to prevent either disease or growth promotion. And finally, I will leave you with two things that I really like. One is a quote and one is a picture. So this is one of my favorite quotes. The universe is full of magical things, patiently waiting for our wits to sharpen. And finally, this is an excerpt from a Calvin and Hobbes cartoon, and Calvin is giving his friend some gyan, and he says, curiosity is the essence of the scientific mind. So my message to you is stay excited, stay curious, adopt the scientific process, be scientists, stay safe. Thank you very much.
Thank you very much, Raghu. There have been many, many questions in the Q&A box, and I'll run through some of them, which I thought are nice if you answered them. I answered some of the others, which I could.
Shall I stop sharing?
Yes, that would be nice. Yeah. So let's start with Sudhanva's question. He asks, are there new non-microbial pathogens too?
Yes, there are. There are eukaryotes that can be pathogenic, and there are microscopic eukaryotes that are part of the human body. And this is still a fledgling field. We are still trying to figure out what these are. There are different kinds of eukaryotes that are present in your body, things like Cryptosporidium, Giardia, various other classes. And they can cause infections under immunocompromised conditions, and they're called opportunistic infections. So technically, we won't call them microbes because they don't fall under the categories of bacteria, viruses, fungi, or parasites. But there are eukaryotic pathogens that are microscopic and cause infection. Yes.
Okay. So there's still
like
Plasmodium.
Like Plasmodium is a Plasmodium is parasitic. It still qualifies as a microbe, but then there are eukaryotic organisms, which I just told you about, that don't fall under these four, but they can still cause infection.
Okay. So there's a philosophical question where they ask, why don't we call the good bacteria present in our body the fundamental unit of life instead of a cell?
Oh yeah, that would be interesting because there are more bacteria than human cells. So we are actually colonizing bacteria rather than the other way around. But this is an argument for another day. But we are still trying to figure out what these bacteria do here. And the hope one day is that there will be, there will be a day when you can actually use these microbes instead of using compounds derived from these microbes. You can use collections of microbes to actually solve or treat human conditions, not necessarily infectious disease related conditions, but conditions that I talked about. You could have mood disorders, you could have weight issues, you could have pain issues. So if you can identify microbes or collections of microbes that are involved in these fundamental processes of human physiology, you could potentially treat them with collections of microbes. But the challenge would be to identify which microbe or which collections of microbes would be involved in certain physiological pathways before we take this into the preclinical phase. But this is an exciting, it is a very exciting area of research. The whole facet or the whole area of microbiome research is opening up now, primarily because methods and technologies and the cost of genome sequence has gone down exponentially. So you can sequence large collections of organisms very quickly and at very low cost. Until a few years ago, it would cost a lot and you would not be able to generate these sequences very quickly. Plus, the advancement in computational biology approaches, once you have these sequences and what we call big data nowadays. So big data analysis is now cannon fodder for people with computational experience. We are now seeking out computer scientists to write algorithms to make sense of the sequences or the data that we generate. It could be genome sequence, it could be transcriptomic sequence, it could be all kinds of systems biology related data. We want people who have computational experience to make sense of this. So this and other big data related health fields are probably the next frontier. And many of you, once you graduate college, would have these as potential career choices.
Good. So somebody is asking, when is it that the immune system cannot stop the bacteria from causing infection?
Excellent question. So like I said, most of the time the immune system actually clears the infection. And so for most of us who are immune competent, which means our immune system is competent and is in good shape, it's able to fight and kill the bacteria. So there are two scenarios under which you could get infected. One is if you're immune compromised, meaning that your immune system is actually weakened by something, either because of a nutritional reason or because you had an infection of something else. Let's say you had a viral infection, which made you immune compromised, and now you are susceptible to a bacterial infection. So under an immune compromised state, you become susceptible to various classes of infections. The other scenario in which you could get infected with a bacterium, even if you're immune competent, is if it's a highly virulent bacterium, okay, or a highly virulent microbial agent. For example, you have this pandemic going all around, and it is, this virus is infecting pretty much everyone, okay? And we're all immune competent, correct? We're not getting sick and falling down. But then almost all of us across the world seem to be equally susceptible to this virus, which means that it has the ability to surmount our immune system or evade our immune system. And that's true for various classes of bacteria as well, including Mycobacterium tuberculosis. So you could be completely immune competent, but you could get sick with MTB just because there was a highly virulent strain and it knew how to outwit even an immune competent immune system.
So the next question is a little bit on history. When arsphenamine was discovered in 1909, why is penicillin then called the first true antibiotic?
Oh, very good question. So arsphenamine is actually a synthetic compound, which was actually synthesized in the lab by synthetic processes, which means it was man-made, correct? So an organic chemist actually synthesized a compound which was tested for its antibacterial activity. So it is an antibiotic, but in the sense of the context of antibiotic being something that a bacterium produces, it falls outside that class. So penicillin is called the first antibiotic because it was the first antibacterial compound that came out from a living bacterium. Okay. Arsphenamine would be the first antibacterial, but penicillin would be the first antibiotic because technically the term antibiotic refers to something that was produced by a bacterium.
Right. Okay. I'm now going to combine two questions. What are phages? And if we introduce phages to our blood, how would it help us?
Oh, okay. Good question again. A phage or a phage is a virus that infects bacteria. Okay? Like of these viruses that infect animals and humans. We have different classes of human viruses. You all know about dengue and chikungunya. Now everybody knows about SARS-CoV-2. But just like animal viruses, there are viruses that infect bacteria. Okay, and these are called bacteriophages. Okay, so wherever you will see bacteria, whether it's in the soil or in water or in air, wherever there is bacteria, there are phages. Okay, and these phages are extremely specific to bacteria. So, certain phages will only infect certain bacteria. And once they infect, like I said, the phage could either lyse, which means kill the bacterium, or it could stay in a lysogenic state, which means it could sit inside the bacterium with its genome integrated into the bacterial genome. So every time the bacteria replicates, the genome of that phage replicates. And eventually, under some certain conditions, that phage could actually come out from that bacteria and start its cycle all over again. So the question is, what would happen if you inject these bacteriophages into the blood? So I presume you're asking me that in the context of treating infections. So let's say you had an infection that couldn't be treated by an antibiotic. The strategy to carry out phage therapy would be to identify specific phages that would kill the bacteria that has infected you, which means you would have to isolate that bacteria, you would have to culture it in the lab, and then you would have to do tests to see if there is a phage or a collection of phages from a larger collection, which would kill that bacterium in vitro, which means in a test tube. Then you would take it to a preclinical and a clinical stage. If it cleared a clinical trial, then you would inject this, either one phage or a collection of phages into somebody who had this resistant infection, which could not be cured by the known antibiotics. And the hope would be that it would clear this infection. Now, phage therapy hasn't become a reality in the clinic, but the methods of testing and isolation of phages are being refined. We are now understanding more about how these phages work. This is a challenge, but then this is a potential non-antibiotic kind of therapy, which would have very low levels of potential resistance.
All right. So some of the students want to understand a little bit better about DNA and plasmids. Can you quickly give them a summary?
Okay, very good. So DNA is deoxyribonucleic acid, which all of you know is two strands of DNA in the anti-parallel conformation wound around each other. Plasmids are actually circular extrachromosomal double-stranded DNA elements that are normally present in many bacteria. Okay, so plasmids, when I say extrachromosomal, it means that they exist outside of the genome. They're inside the bacterial cell, but they're not integrated into the genome. So every time a bacterium replicates, the plasmid also replicates. And you can have different kinds of plasmids. We call them low copy number and high copy number plasmids. So you could have more than one copy, which means one bacterial cell could have more than 5 or 10 copies of that plasmid. So what is the use of this plasmid? Why do bacteria actually have plasmids? So bacteria have plasmids presumably because it confers some kind of survival advantage in their natural environment to the bacterium. It could be either in terms of encoding some kind of DNA element which provides it protection from other bacteria that is trying to kill it. Okay, so these plasmids are all pervasive. They're present across the earth in all kinds of areas. It could be soil, water, wherever bacteria are present. They would have, many of them would have plasmids. And if you wanted to know what the identity of those plasmids is, you would have to isolate them from the bacterium, sequence the plasmid, and see what elements they have. And many times when you do this, you find that these plasmids have what we now know as antibiotic resistance cassettes. And these plasmids can be transferred from one bacterium to another because bacteria are naturally competent, which means that they have the ability to acquire these either plasmid or other DNA sequences from their environment naturally. So this is one way by which bacteria become resistant to antibiotics, by acquiring plasmids that have antibiotic resistance elements on them.
Okay. This is an interesting question. Common cold is a disease that has been around for quite long, but still there is no vaccine for it. Why is that so?
Okay, so now we are going to the domain of viruses. We know common colds are caused by viruses, flu viruses, the class of influenza. Now the problem with viruses that cause the common cold is that they mutate very quickly, which means that they can develop a huge number of variants in a very short time. So if you developed a vaccine against one particular variant of this common cold virus, by the time you send it to the clinic or you send it to the population, that strain of virus has already mutated into something else. So because viruses have this amazing ability to mutate and make variants, vaccine development is a challenge. And you see how much of a challenge vaccine development is, even in the context of SARS-CoV-2 infection. You have the vaccines that are currently in circulation, the ones that many of us have taken, don't always protect against the variants that are constantly arising. So that's a challenge. And the reason is because viruses mutate quickly and generate variants. And because of selection pressures, certain variants are fixed in the population, like this, the Delta variant or the Omicron. They are variants that were actually, they had a survival advantage or a transmissible advantage or a virulence advantage. And that's why they're fixed in the population amongst the hundreds of thousands of variants that may have been around. So developing a vaccine against one variant may not necessarily protect against another, just because vaccines are specific to certain components of viruses, and viruses have the ability to develop mutations and change those components.
All right. Now let's move to allergies. Can we say that allergies are also diseases?
Okay. So this is an allergy would be termed as a condition. I would term an allergy as a condition and not as a disease, but people with allergies will call them diseases because it makes them. See, the word dis-ease or disease comes from dis-ease, which puts, anything that puts you out of ease is a disease. So in that sense, an allergy is a disease. It's not an infectious disease, but many times allergies are because of a hyperimmune response to something, okay? And we discussed the immune system to be an integral part of response to pathogens, but then sometimes the immune system is responsible for different kinds of other diseases, and one of them is allergies. So an allergy is essentially a condition where a person develops an immune response to something that not everybody responds to. It could be pollen, it could be dust, it could be anything, a fragrance or a smell or an aroma. So an allergy would be a disease, yes, in the definition of the term disease, and it involves the immune system.
All right. So I'll take this as the last question because it's almost 4:30 and we need to start with the selection test after this. And for all the participants here, after this question, we will tell you how to log in for the test. So stay with us. So the last question is by Amog. Is there anything that targets bacteriophages? And is there a possibility of phages crossing over into humans like other viruses have done?
Oh, thank you, Amog, for that question. No, bacteriophages don't infect human cells, and those have been tested. Okay, bacteriophages are called bacteriophages because they only infect bacteria. And the way they work is, these phages attach to something on the outside of the bacterium, okay, and which is a component of the bacterial cell wall. So there is attachment between a component of the bacteriophage, usually some protein in its tail, and that component binds to a component on the bacterial cell wall. And those components of the bacterial cell wall are missing in human cells. So essentially points of attachment are missing. So bacteriophages infect bacteria and not animal cells. So the possibility of crossing over is unlikely. Although you never know, you never know what happens when you have long-term effects of therapy. But to the best of our knowledge, bacteriophages don't infect human cells.
All right. Thank you very, very much, Raghu, for patiently answering all the questions. And thank you all the participants here for putting such lovely questions. Pity that I couldn't address all of them, but I'm sure that we'll have many other opportunities to interact with Raghu or other scientists to have them clarified. As we move on, I'll request Ankaj from Exam Online to quickly demonstrate how should participants participate in the online test for the YIP. Ankaj,
So I should say bye-bye?
Yes, Raghu.
Okay, thank you all for being here and listening to what I said. And I hope there'll be a chance for us to interact in person sometime soon. So although this is an unusual setting, I don't really like looking at the screen all the time and not seeing any of you, but I hope you are energized and excited and curious about the things I talked to you about. So thank you very much, and take care, all of you.
Thank you so much. Pankaj, over to you.
Hello, ma'am.
Yeah.
Hello, everyone. I'm looking for your exam today. So first of all, I'm telling about how to log in, how